In this lecture, we're going to examine another very important macromolecule, and that is protein. Now, proteins are very important. In fact, the term protein, it means of prime importance. These are organic molecules, which is to say that these are carbon-based molecules and are the second most abundant component of plants and animals, only second to water. And as just indicated, these are macromolecules, and these are macromolecules involved in all biological processes and are central to understanding human physiology. From a dietary perspective, protein is available in a wide range of foods, both animal-based foods and in plant-based sources. Now, the amount of protein available in these foods differs, as well as the types of proteins between these different foods. And so that suggests the idea that not all proteins are made alike. So some of the key properties of proteins, just like we saw with carbohydrates, proteins are macromolecules, which means that they're polymers that are chains of individual monomers. So where the monosaccharide was the monomer for carbohydrates, the amino acid is the monosaccharide for proteins. And when proteins form, or when amino acids are put together in a linear chain, that linear chain is then folded and twisted in a number of different ways to form a 3D structure. The specific type of 3D structure will eventually dictate the function of that particular protein. Proteins contain a wide range of different functional, or what are called chemical groups, and so these are just examples of some of those chemical groups. And chemical groups are collections of a small number of atoms that will define the properties and characteristics of that particular molecule. And so these are just some examples. We have an OH group, an SH group, which is sulfur and hydrogen, an NH2, and a COOH. And we'll come back and we'll see examples of these. And these will allow for chemical reactions. And when we talk about chemical reactions, these involve either the formation or the breaking of chemical bonds between atoms, as well as between molecules. And so proteins interact with other proteins, as well as other macromolecules, particularly carbohydrates and lipids. And so these will form larger assemblies. And so just some examples here that we'll see a little bit more specifically in a subsequent video is related to cholesterol. Now, there's really no such thing as good cholesterol or bad cholesterol, but it's a term that's used in public health to describe really what we are referring to as a lipoprotein. And so by definition, the lipoprotein is a collection of lipid molecules and protein molecules. And these are globules that circulate through the body, and some lipoproteins contain a higher amount of cholesterol and some contain a lower amount of cholesterol. And that's what gives those particular lipoproteins the term good or bad. But cholesterol is defined by chemical structure that is actually necessary for a number of physiological functions. Proteins can either be rigid or be flexible, but that is limited. In a subsequent video, we'll take a look at the cytoskeleton. And the cytoskeleton is a collection of proteins in the cell that acts as a scaffolding or a network that provides that strength and support and gives cells particular structure. However, the cytoskeleton is unlike a house or a building. The cytoskeletal framework is flexible, so it can bend, it can twist, and that's what allows cells to operate optimally by deforming. But that is limited, so you can bend a cell or deform a cell to the point where the cell will fracture. But what these proteins will do, it will allow the cell to deform up to a point without fracture. And so the terms levers and springs are terms that are often used to describe the characteristics of some cells. So when we talk about an amino acid, we begin with the central carbon atom. And so amino acids are often referred to with respect to human physiology as alpha amino acids, centered at what's called the alpha carbon. And so we take a carbon atom, and we attach a hydrogen atom to it. Then on this side, we're going to attach a functional group called an amino group. An amino group is defined as a nitrogen atom and two hydrogen atoms chemically bound together. On this side of the carbon atom, we're going to add what's called a carboxylic acid group. And a carboxylic acid is defined as a collection chemically bound together of one carbon atom, one oxygen atom, another oxygen atom, and a hydrogen atom. Now, all amino acids, and with respect to human physiology, there are 20 amino acids. All 20 have this identical structure that you see here. And so we have an amino group on one end, a carboxylic acid group on the other end, and the term for the entirety of the molecule just borrows terms from these two functional groups, and therefore we call this an amino acid. Now, what differentiates the 20 amino acids from each other is this down here, and this is what's called a side chain, or an R group, and also referred to as the variable side chain. So R is not an element. R is a placeholder where we can have a wide range of different atoms or different groups of atoms placed here, and that will differentiate one amino acid from another amino acid. And it's important, once again, here, this is an organic molecule centered at what's called the alpha carbon. And so amino acids differ by the R group, and so the R group is just a placeholder, and that gives rise to 20 different possible side chains. And depending upon which atom or which group is added to the side chain, that will dictate the size of the amino acid, the shape of the amino acid, the charge, the ability to bind hydrogen atoms, how scared that atom or that molecule, that amino acid, is to water, which is the definition of hydrophobic. So some amino acids can dissolve in water more easily than other amino acids, depending upon what's in that side chain. And some amino acids will form chemical bonds with other molecules easier because of that side chain, and that's what chemical reactivity is. All proteins are made from the same set of 20 different amino acids, and these are the amino acids that have existed for several billion years. There are two general classes of amino acids. The first are referred to as the essential amino acid, and out of the 20, there are 9 amino acids that are considered essential. It is very important at this point that you do not confuse the term essential with important. All 20 are important from a dietary perspective, but the 9 are essential, which means that the human organism cannot synthesize, cannot make these 9 amino acids, which means that we must take these amino acids in from the diet. The remaining 11 are considered non-essential, which means that the human organism, primarily our liver cells, can make these amino acids, or are conditionally essential, which means that the human organism can make these amino acids on the condition that we consume an essential amino acid. These 11, the human body does have the capability of synthesizing, but are still considered critically important for normal human physiological function. If we take a look at the 11 non-essential or conditionally essential amino acids, we see that if you take a look across these chemical structures, you see they all share the same characteristic. right? So they all have the amino group, the carboxylic acid group. This is illustrated a little bit differently than I had on the previous couple slides, but they all share the same basic core atoms, with the exception of the blue. So we'll start here at the top left-hand corner. We see that if we take a single hydrogen atom, one hydrogen atom, and put it in that R, and replace it and put it where the R was. Remember, the R is not an element, it's just a placeholder until we stick something there, and we're going to stick a hydrogen atom there. This, by definition, is now called glycine. Glycine is a non-essential amino acid. Humans have the capability of making it, and it is considered the simplest of all the amino acids because it's the simplest chemical complex. And you can see here with these other side chains a wide range of different complexes that gives these different amino acids much greater flexibility in terms of chemical reactivity with different molecules. The next complex, or the next, or often you can think of it as the next simplest or the next complex, is alanine. And so alanine is defined by a carbon atom and 3 hydrogen atoms in one group placed in that variable side chain. And CH3 is referred to as a methyl group. So these different side chains, these different chemical structures are not necessary to memorize, but it's important to note how these amino acids are different from each other. We see the essential amino acids. Once again, we're talking about 9 of the 20 humans have to ingest in the diet in order to obtain these. Now, 3 of these that I want to highlight here is isoleucine, leucine, and valine. And these 3 unique essential amino acids, now when I say unique, they fit a chemical definition of this branched chain amino acids, in that the chemical properties of these variable blue side chains are considered branched chain. They're different from each other, but they share a common feature in terms of the branching nature of the additional carbon atoms that exist inside those side chains. Now, the reason the branched chain amino acids, isoleucine, leucine, and valine, are unique, that these 3 amino acids account for approximately 1/3 of all amino acids in muscle protein. And so that's a pretty sizable number when you consider muscle protein is made up from a combination of 20 different amino acids. So there's a large array of amino acids. 1/3 are composed of just these 3 amino acids, indicating the importance of ensuring the human diet contains enough protein that will deliver these essential amino acids, particularly the branched chain amino acids. What we're looking at on this slide are common computer-generated depictions of the structure of different proteins. So you can see some proteins you can detect somewhat of a symmetrical shape to it. Other proteins do not appear to have any sort of symmetry to that particular molecule. But you can see these complex 3D compilations, and these are made up of only amino acids in a wide range of different combinations. What we see here in the upper left-hand corner is a protein called hexokinase. And hexokinase is an important protein that functions as an enzyme. We know it functions as an enzyme because of these last 3 letters here. So any term you see with ASE, with the suffix ASE, indicates that A, it's a protein, and it's a protein, B, that functions as an enzyme, which functions to speed up a chemical reaction. On the right-hand side, upper right-hand corner, here we see a different protein. This particular one is human pancreatic lipase. Once again, ASE, this is a protein, A, that B functions as an enzyme which speeds up a chemical reaction. This particular protein is derived from humans, so humans make this in the pancreas, and it speeds up the breakup of lipids, dietary lipids. So this is an important enzyme involved in the digestive process. Ovalbumin here is the major protein, the most abundant protein found in eggs from a dietary perspective. So you see a very complex chemical structure here in ovalbumin, which makes it somewhat of a challenge for the human digestive system if the person consumes eggs to break up. This is important to denature this protein from a dietary perspective so the human can absorb the amino acids. And that's why cooking eggs is an advantage, because the cooking process uses thermal energy to begin the process of breaking up the chemical bonds holding this protein together. That will pre-digest the egg protein, and then the human digestive system has an easier time breaking it up. Beta-lactoglobulin, here's a different sort of computer reconstruction of beta-lactoglobulin. This is the most abundant type of whey protein, WHEY protein. So there are two types of protein in cow milk. The most abundant protein are what are called the casein proteins. That makes up about 3/4 of all protein in milk. The other 25% are this general family of proteins called the whey proteins, and the most abundant whey protein is beta-lactoglobulin. So proteins carry out a number of very important functions in terms of human physiology. The first function here is mechanical in nature. So when we think about tissues such as muscle tendons, skin, ligaments, these are tissues that provide structure and support. And so an important protein involved in that is collagen. And collagen does provide that structural strength. There are other types of proteins that we'll touch on throughout the course, and these are called biomolecular motors. Myosin is an important biomolecular motor which is found in muscle tissue. It's actually found in all cells, most cells, but it's found particularly in muscle cells. Kinesin and dynein are found inside neurons, and these all perform some sort of motor action, hence the term biomolecular motor. As touched on throughout this lecture, enzymes are proteins that function to speed up chemical reactions, and these are critical to human physiology, and we'll touch on these periodically in subsequent videos. Hormones are important chemical messengers released by glands that carry out a wide range of functions in the body. Insulin and glucagon are functionally hormones, but structurally are proteins. And these are both produced by cells in the pancreas. In terms of immune function, proteins are very important. Antibodies, which are produced by a particular type of white blood cell called the lymphocyte, are actually structurally proteins. And these are blood proteins that function as part of the immune system to neutralize the action of contaminants and will bind to what are called antigens, which are components of viruses in an attempt to neutralize those viruses. Proteins in the blood, albumin is the most abundant protein in human blood that's found in plasma. Globulins are another type of protein found in blood, and these proteins set up what's called an oncotic pressure and will help to regulate fluid balance, not just in the blood, but also in extracellular tissues. Proteins are important in maintaining acid-base balance, and so in order to maintain a slightly alkaline blood level, blood pH of 7.4, blood proteins are effective in helping to bind atoms or molecules that would either drive that fluid to be either more acidic or more alkaline. So it's important that the human bodily fluids, both extracellular fluid, intracellular fluid, and blood, maintains that optimal pH level. As we'll discuss much later, proteins serve very important cellular transport functions, and we're going to speak specifically to what are called the ATP aces. These are proteins embedded in various cells in the body that actually cost money to operate. When I say cost money, we'll break up ATP in order to transport ions such as sodium, potassium, and calcium. And last, in terms of protein function, is that proteins can be broken up in the body to get the amino acids to run through a metabolic process called oxidative deamination, and as a result can serve as a source of energy to make ATP, and that is the last of the macromolecules that we want to use for energy. For the body to burn protein to provide ATP, it's going to have to sacrifice and break down muscle tissue, and that's what occurs in advanced and later stages of starvation, and that's why consuming dietary carbohydrates will help to protect muscle tissue, because if we don't consume dietary carbohydrates, the human body will start to break up muscle protein to get the amino acids to not only burn those amino acids for ATP, but to convert some of those amino acids into new glucose molecules, and so that red blood cells and neurons can use those glucose molecules. Excess protein intake from the diet can be converted to fatty acids, but this is not a metabolic pathway that humans use abundantly, but is usually a pathway that will activate when the human diet is in an energy surplus. And so the body is consuming more carbohydrate than the body can possibly use or store. So this ends our lecture on protein.